think we'll get started. So it's a pleasure today to welcome Dr. Kim McAllister from UC Davis. As it turns out, I just found out talking to Kim, I guess this morning, when she was originally recruited as a faculty member at UC Davis, Mark McNamee, our provost, was the person who recruited her. And so that was sort of Mark's last year, I guess. He recruited Kim and then figured that was his crowning achievement, and he moved to Virginia Tech that year.
Or we scared him off, I don't know.
Right. Anyway, so that's a great connection to Virginia Tech I didn't appreciate. Kim did her undergraduate work in North Carolina at Davidson University and then went on to do her PhD also in North Carolina at Duke. She was a student of Larry Katz. Many of you, I'm sure in this room, remember Larry Katz, who died sadly and tragically at an early age, who was really one of the great pioneers in neurobiology. And Kim had the opportunity to do her graduate work with him. From there, she went out to do a postdoc at the Salk with Chuck Stevens. And then from there, she joined the faculty at UC Davis, where she is currently a professor of neurobiology, physiology, and behavior, and of neurology. Truly a professor in two different departments. She also serves as the associate director for the Center for Neuroscience at UC Davis, which again I'm sure many people in this room know is a really outstanding program. Kim has a number of types of recognition for her accomplishments. She's been a NARSAD established investigator. She won the Society for Neuroscience Young Investigator Award. She serves on the Scientific Advisory Board of Autism Speaks and serves on a variety of editorial boards, including being a reviewing editor for Frontiers in Molecular Neuroscience. When she started out her graduate work with Larry, she was working at the time on neurotrophins, and that was really an exciting early days of neurotrophins and a whole field that's grown out of that, and really gave some of the initial important insights on the role of the different neurotrophins and receptors and signaling pathways in elaboration and development of structure within the brain, within the cortex, and did a series of really nice studies that separated out specific effects of different neurotrophins like BDNF and NT3 and so forth, and their effects on the development of dendrites, particularly interesting to me anyway at the time, following different dendrites, different domains on a cell and the different effects of the neurotrophins, which again really opened up a very important area. In Chuck Stevens' lab, she of course was doing physiology and quantum analysis and was involved in an important series of studies there. And I think the graduate students in the neuroscience course will resonate with this, to understand the saturation of receptors, glutamate receptors, by release of a quanta, and that you didn't necessarily have full saturation, and the implications for that of our understanding of quantal synaptic transmission. She went on to do a whole series of studies understanding the trafficking and the position and the particles that give rise to the placement of glutamate receptors, particularly NMDA receptors, and how they move with the plasma membrane during pauses in trafficking and so forth. And over recent years, she's really focused her work on understanding the interaction of molecules from the immune system, particularly the major histocompatibility complex 1 family of molecules, and neuronal development and plasticity, and identifying and understanding these signaling pathways and what it means for brain development, and also in pathology. And she's got I think, a very interesting, fascinating story about the potential role of these pathways in neurological diseases such as autism and schizophrenia, and understanding and identifying some novel pathways and interactions between the immune and the nervous system. So really look forward to hearing Kim's talk today. Welcome, Kim. It's great to have you here.
Thank you so much. So that was such a nice introduction. Thank you. I don't know that there's anybody else who knows the breadth of the things that I've done. So that was really impressive. And you know, I want to, before I get into things, thank you for the invitation. I have not been to Roanoke before. I'd heard bits and pieces watching people get recruited to your institute, and it's really quite impressive. You guys are lucky to work here. The facilities and the faculty are awesome. And I'm sure the students and postdocs will be when I meet you guys later. So in general, there is a theme to the broad number of things that I've done over the course of my career, and that really is trying to address a very, very general question, which is, how do connections form between neurons in the developing brain, and how are those connections modified by activity, by experience during typical brain development, and then also by disease in neurodevelopmental disorders? And the areas of the brain that I've focused on in the past and that Mike mentioned are mostly here in the back of the brain, the visual cortex, and most recently we've started to delve into the front of the brain, looking at development of prefrontal frontal cortex areas. The neurons within the regions of cortex that we study are typically the primary excitatory neurons, these pyramidal cells that are found throughout the 6 layers, other numbers of layers in other brain regions. And these cells are completely covered with synapses. They have thousands to tens of thousands of synapses all covering their dendritic arbors. And there are many different cell types on these cortical neurons, but the synapse types that we study the most are glutamatergic synapses, which are the primary excitatory synapses on these cells. And those synapses have a really typical ultrastructure, which you can see here on the left in this electron micrograph. All of you are probably familiar with this. This is the presynaptic terminal that's filled with synaptic vesicles. It's tightly opposed to a postsynaptic density within the dendritic spine. That's this fuzzy stuff. And over the last 10 years or so, 10 to 15 years, there's really been an explosion in our understanding of the molecular complexity of these synapses. And the diagram on the right is, I think, an incredibly oversimplified diagram of what those protein-protein interactions might be and what the protein composition of these synapses are. It turns out that recent estimates from Seth Grant's group using proteomics approaches have identified over 1,000 different kinds of proteins at excitatory glutamatergic synapses. So as we start to appreciate that incredible molecular complexity, to me as a developmental neurobiologist, it becomes that much more daunting to try to imagine how those hundreds of recruited to new sites of contact between axons and dendrites to even form one synapse properly. These have to be recruited in the right hierarchy with the right timing in order for synapses to function and to have their plasticity that we know and love, and that's important for brain function. But when you think about it on the flip side, the whole process of synapse formation is absolutely critical to brain development. We have to have this happen properly. So we should know a lot about it, and we don't. And so when I started my lab about 15 years ago now, which is a really long time, I don't know how time flies so fast, it really was amazing to me that we didn't understand this basic process that's so fundamental to all of us. And there are a lot of reasons why we don't understand a lot about synapse formation, and one of them is just a really simple fact about scale of synapses. So a synapse is on the order of 0.5 to 1 micron, sometimes much smaller in the developing brain in diameter, and any millimeter of tissue contains about 1 million neurons and 1 billion synapses. And so it's easy enough for us to imagine a cubic millimeter of tissue. It's impossible for us to imagine this kind of complexity and this kind of packing density. And so it has been really difficult to watch synapses as they form in the intact brain for this reason, especially as they undergo their initial formation in the cerebral cortex, which doesn't occur on spines. It occurs along the shafts of dendritic arbors. And so it was about 15 years ago that techniques that allowed us to take neurons out of the brain and culture them and then watch them grow in culture were developed. And this really opened up the field of being able to study the dynamics of proteins as synapses form. And so this is a figure that's taken from the Culture Bible. It's from a book by Gary Banker, who really was the founder of this field. And it shows these little neurons. These are hippocampal neurons. Cortical neurons look just the same. When you take them out of the brain, they're small. They've got cell bodies, some minor processes that will turn into dendrites, and then axons that extend out and are tipped by these growth cones. And so if you plate them at low enough density, you can find times after plating when the neurons are starting to differentiate but have not come into contact with each other yet. And then over subsequent days, you can start to get a sense that they're forming many, many connections because these networks become very complex. So after about 3 weeks, you can see that these are the cell bodies, the darker gray areas here, and all of these lines, these little tracks in between them are dendrites, and they're covered with axons, and if you stain for synaptic proteins, the synapses are incredibly dense all over the cells, all over the surface of these cells. So to give you a sense of how these cells grow, I want to show you a video from a new assay that we've developed in the lab where we, in this case, have transfected all of our cultured cortical neurons, and these are from cultures that are made from newborn rat pups from visual cortex. And all of the cells are transfected with a lentivirus that expresses GFP in the cells from 4 days in vitro to about 8 days in vitro. So they're transfected at right before the synapses are starting to form in these cultures. And so all of the cells are green. They're all labeled with GFP. They're all white here. These are the cell bodies. There's a clump of cell bodies over here. The thicker processes are dendrites, and then the really thin processes that you're going to see here are axons, and what you'll see is that they're tipped with growth cones and that a lot of the growth is happening as the axons are extending. So this new long-term imaging assay is one where we can image every hour for 4 days. And so the time scale that you're going to see up here is hours and then days. And I think that this video is really fun because it gives you a sense for what's actually happening in these cultures. The process is incredibly dynamic. You see a lot of extension of dendrites and large branches that are extending across the culture dish. You can also see quite a number of thinner processes, axons that are growing across the culture dish and that are tipped by these really amazing growth cones. You're going to see one come in here and see that? And then it's got the really neat growth cones. There are also really interesting dendrite dynamics that you can see here. There are dendritic branches that extend off and then are retracted, and all of them are tipped with these beautiful growth cones again. So using this kind of an assay, you can not just label cells with GFP, but you can also label them with pre- and postsynaptic proteins, and then get a sense for what the distribution and dynamics of synaptic proteins are before and then during synapse formation, and we hope using this new assay during synapse elimination and synapse dynamics as networks mature. So using this kind of technique, I'm going the wrong direction. There we go. So there have been a number of advances that we've contributed some to and that have been made by other people in the field that have led us to believe that synaptic proteins are absolutely found in axons and dendrites before synapses form, and they're found in transport packets that are highly dynamic in those cells before synapses are formed, and that undergo interesting forms of regulation that the field is only just now beginning to appreciate. So the way that we know this is that we can transfect cells with a synaptic vesicle protein that's fused to GFP or to a red fluorescent protein, M-cherry, or we can transfect cells with NMDA receptor subunits, in this case NR1, that's fused to a green fluorescent protein again, or M-cherry, a red fluorescent protein. And we can take movies of the mobility of these cells. And for this example, all of these cells had not made contact with postsynaptic partners yet. So these are cells that do not have synapses, and these are synaptic proteins, but they're not at synapses yet. They're found in these processes before synapses are formed. And you know, this, I want to make a note about this because some of you are studying synapses and you're studying it in synapse development. What I think this data really shows us is that if you can't define a synapse by either pre or postsynaptic proteins, because the cells are really full of these. You really have to see where you have co-localized pre and postsynaptic protein in order for that to be defined as a synapse. And so the kinds of dynamics that we saw were really surprising. They're really unexpected. This movie is taken with a different time scale than the one that I just showed you. So this is an axon from a cell in which all of the clusters are synaptic vesicle transport packets. The imaging is taken frames are taken every 10 seconds for a total of 10 minutes, and the video is looping. And so when you see it flash, that's when it goes back. There are some stable puncta, but there are also a ton of these that are moving around, and we've quantified this mobility. They move in both anterograde and retrograde directions, and they pause, and those pause sites seem to regulate where at least the initial complement of synapses will form onto these neurons. Yep.
Are these packets passing each other in a given branch, or is it a one lane?
Nope. So they're absolutely passing each other. So even within the axon that's pretty thin, there are multiple microtubule tracks. As we start to develop techniques for super resolution imaging that can be applied on a rapid time scale to living cells, I think maybe we might be able to start seeing the different tracks and figure out, you know, why you have this kind of heterogeneity, and what the transport packets are doing when they pause and how they hop back onto different tracks. Because it may be that there are different properties of mobility of these transport packets on different microtubule tracks. But no, I mean, at the resolution that we have, this looks like a really big cluster that should just fill the axon. It's not, because they bloom a little bit, and they're much smaller than what we can actually see with our light microscope. So we see similar kinds of mobility of NMDA receptor, let me find my mouse, there it is, transport packets in dendrites. And so this is a cell that's transfected with NR1 M-cherry. The cell body is actually right here, and these are the dendrites. And so there are many fewer of these mobile puncta, but they are absolutely mobile, and they move around both anterograde, retrograde directions and pause. So we think that these transport packets are actually important for synapse formation because we see them recruited to new sites of contact relatively rapidly. And so one movie of this, and then I'm going to summarize a lot of the data that we have on this. The movie on the left is really, I think, an incredible video that was developed by a former postdoc in the lab, Steph Barrow, who was just amazing at this kind of imaging. This is pretty difficult to do. And again, the frames were taken every 10 seconds for about, I think, an hour in this case, because the likelihood of a growth cone actually making a connection with the neighboring dendrite is pretty low, and it takes a while for it to do that. So this is a growth cone from a cell that's transfected with red fluorescent protein, so it's just labeled red. This is a dendrite from a cell that's transfected with neuroligin GFP. Some of you in the synapse field know that neuroligin is one of the most well-studied and probably one of the most important molecules in inducing the formation of excitatory glutamatergic synapses between cortical neurons. People had made those conclusions based on a number of studies that manipulated the protein and then looked at later consequences on synapse density, but nobody until Steph did seen whether neuroligin was found at the right place at the right time when synapses are initially forming in order to be able to regulate that process. And so that's really the power of this kind of approach. And so what you're going to see is that this growth cone is going to come down. It's going to actually cross over the dendrite. And so watch what happens to the green there. You'll see a really rapid accumulation of a cluster of neuroligin and then some interesting behavior. So you see that rapid accumulation, and then there's a phylipodium that actually extends up the axon from that dendrite. And we see this behavior a lot. It has not been described at all in intact tissue. We don't know whether this is something that's specific to culture. That could be. A lot of what we see in culture ends up reflecting what we see in vivo. And what we think is happening here is that there's an extension of a phyllopodia that may be neuroligin dependent. Certainly, the cluster of neuroligin happens almost immediately, and there's a cluster of neuroligin at the tip of that phylopodium. And we think what's happening is that is enhancing adhesion between the pre- and postsynaptic partner to increase the amount of time that they're adherent, which allows for more time for the synaptic proteins to be recruited to those sites. Yeah, so when you knock down neuroligin, you get many fewer contacts forming. And so using an imaging assay like this, when your likelihood of actually seeing a contact form is relatively low in the first place, to see fewer contacts form is not going to be something that anybody wants to do, because it would take months and months and you still wouldn't actually know whether you had a real effect there. So certainly in cultured neurons, when you knock down neuroligands, you get a decrease in synapse density. Whether this behavior is important for that is pretty difficult to determine. We can't do it by manipulating actin either, because then we don't get the extension of the axon, and you don't get the accumulation of the proteins there either. So it's a tough question. When we look at other molecules, really rapidly within seconds to minutes at new contacts between axons and dendrites, and that's followed by the recruitment of synaptic vesicles in the presynaptic terminal and NMDA receptors on the postsynaptic side within about 7 minutes, with a similar time course, within about 7 minutes to new sites of contact. And then later you get accumulation, it appears, of a lot of the other molecules. So that's really led us to the current model of synapse formation, which is that synaptic vesicles and postsynaptic receptors are found in mobile transport packets before synapses form. Contact leads to some signaling event that we don't know about that leads to the rapid accumulation of neuroligin and some sort of a bidirectional signal that causes accumulation within minutes of synaptic proteins. In the lab, we're working out a number of these signaling molecules, and we were talking about trying to get papers out this morning. We have a bunch of papers in hopefully that are on my desk in final stages. It'll be out pretty soon, looking at the role of actin and some kinases that are involved in trafficking and accumulation of these molecules. But in addition to trying to figure out how this happens and what the timing and hierarchy is at individual synapses, there's an equally interesting, maybe more interesting, set of questions regarding this process. And that is what determines whether, where, and how many synapses will form on neurons. right? This is really the crux of how does circuitry form properly in the brain. And there are a couple of pretty obvious answers to this. One possibility is that the presence and distribution of synaptogenic molecules guides whether and where synapses form on a neuron. So many of you have probably heard about synaptogenic molecules. They're called that because they're important for the genesis of synapse. They promote the formation of synapses. And this is a very, very long list of molecules, only some of which I have on this slide. I can't fit any more names on there, but it seems like every couple of months now, a couple years ago, it was every month there was a new molecule, Mike, you know about this, that have been identified that promote the formation of synapses in some region of the nervous system. And these fall into a number of different categories: diffusible molecules that can come from neurons or from glia, and transsynaptic adhesion molecules, of which neuroligin is the founding member, one of the founding members. And this list is the one that seems to be increasing, as well as the glial-derived factors. So there's no doubt that this possibility is absolutely true. Synaptogenic molecules absolutely do play a very important role in the formation of synapses. But there's an equally interesting possibility, and that is that there may be molecules that fall into the opposite kind of functional family, that there may be synapse-limiting molecules whose presence and distribution also may guide where synapses will form and when they'll form on a particular cell. And it turns out that this is a much shorter list. It may be shorter because there are fewer of these, or it may be that we're just beginning to identify this family of molecules. It was initially populated by a report, I think it was in Science, by Mike Greenberg's group in 2006, showing that MEF2 transcription factors limit the formation of connections onto hippocampal neurons. And there have been a number of reports since then that have identified a handful of these molecules. Some of them are sort of typical transsynaptic molecules that act in axon guidance and synapse formation, but many of them fall into an interesting class of molecules, which is immune molecules. So these are molecules like complement and MHC1 that were initially found and are well characterized in the immune system, and that we're beginning to understand or also play a role in typical brain development. And so for the rest of the talk, I'm going to tell you about some work that we've done in showing that MHC class 1 molecules are synapse-limiting molecules, that they're important for the initial formation of connections in the typically healthy developing brain, and that they may actually be mediating the effects of both genetic mutations and environmental influences in autism and schizophrenia. So as many of you know, MHC class 1 they're molecules that are found on all nucleated cells in our body, and they mediate adaptive immunity. They're trimeric proteins that are found in the membrane of all nucleated cells, although they were classically not thought to be found in the brain, which I'll get to in a minute. They're comprised of a transmembrane heavy chain shown here in blue, a light chain called beta-2 microglobulin shown in red, and then they present peptides up here in the peptide binding groove. And those peptides are derived from proteolysis of intracellular proteins. The peptides are loaded onto the heavy chain that's then associated with beta-2 microglobulin, and only with those associations is MHC exported out of the ER and then presented on the surface of the cell. So why did we get interested in this? We were inspired by some beautiful work that came out of Carla Schatz's group about 15 years ago now. So this discovery was one of those things that happens in the lab that you're totally not expecting and then changes the course of your research. And this absolutely, I think Carla would say, in her lab. So as many of you know, the Schatz Laboratory is one of the leaders in studying the mechanisms of activity-dependent development of the visual system. And about 15 years ago, people were starting to think about what the molecular mechanisms might be that mediate that competition. And Carla's group decided to use a new technology at the time, or one of the most used technologies at the time, which was called differential display, which is kind of laughable now because we don't use that anymore. But Rod Corriveau and Jean He, who were postdocs in her lab, used differential display, and they thought they were going to find a whole bunch of activity-regulated genes, and instead they kept finding MHC class 1 molecules. And to hear Rod tell it, he really thought that his career was over. He couldn't do biochemistry anymore. He couldn't do any of this because MHC is not supposed to be found in the brain. And for many, many years until these reports, nobody thought to look for MHC, and when they did, they hadn't found it. And so people didn't believe this until Carla's group started to do some really, really important experiments, and that was to use in situ hybridization to look at mRNA expression of a number of the different MHC class I molecules. And so this is a picture that was on the cover of Science in 2000 that basically shows a coronal section through the brain, and it's the distribution of 3 different MHC class I molecules that are color-coded in blue, red, and green, I think here. And so you can see that they're found everywhere throughout the nervous system, and they can be found in region-specific manners. So they're there, but that doesn't necessarily mean that they're functional. What the Schatz group then went on to do was to show that mice that are deficient in MHC expression, these beta-2M TAP knockout mice, fail to undergo normal activity-dependent refinement of the retinogeniculate projection. And so in mature animals, in wild-type animals, when you label retinal axons and you look in the LGN, you see this beautiful projection pattern that's nice and tight and sort of oval. In the animals that don't have MHC, the projections go everywhere. And so the interpretation here is that there's a deficit in synapse elimination that MHC normally regulates. And so my lab, inspired by this, wanted to see whether MHC might be present much, much earlier in the developing cortex and whether it might regulate the initial formation of connections in the cortical areas. And so a postdoc in the lab, Lee Needleman, did some biochemistry and some really beautiful immunohistochemistry and electron microscopy to address this question and published the paper in PNAS a few years ago. What she used was an antibody that recognizes most MHC class I molecules. This is called OX18. And the first thing she did was to probe light membranes from the visual cortex from rats at a number of different postnatal ages. And these are important ages because they correspond to right after birth, the period P9 when connections are first forming within the visual cortex. P23, this is a period of plasticity, activity-dependent plasticity, and then in the adult. And what she found was that contrary to expectations, which were that we would see MHC only coming up around the period of plasticity, we actually found MHC in these light membranes at much higher levels soon after birth and during this period when connections are first being established. It's still there during periods of plasticity and the adulthood, but it's at much lower levels. When Lee went on to look to see what its distribution was, she found that it's found all throughout the 6 layers of the cerebral cortex at all 3 ages that she looked at. And it's found at high levels within the cell body, within the dendrites, and it looked to be pretty punctate within the neuropil, which is suggestive of synaptic localization. But of course, you can't say that those are synapses unless you do electron microscopy. And so she used both pre-embedding and post-embedding immunoelectron microscopy to look for the localization of MHC. And so this is a representative synapse from a P7 section through layer 5 of visual cortex and an adult synapse. And the gold particles that you see here and here are labeling MHC. And you can see that it's found both pre- and post-synaptically. It's even found right within the synaptic cleft. And that's true for both ages. So we know that it's at synapses, but we were wondering whether it might be on axons and dendrites before synapses form in order to play a role in that initial establishment of connections. And so a graduate student in the lab, Marianne Glynn, used those cultured neurons that I told you about, where we can get really nice resolution of pre- and post-synaptic localization of these proteins. And she labeled them with the OX18 antibody at a number of different ages before, during, and after the peak period of synapse formation in these cultures. And as you can see, there are little green spots at every age in all of the different panels, which means that MHC is found on neurons before, during, and after synapse formation in both dendrites and in axons. And so we still weren't quite sure that this was something we wanted to study, and so we thought, well, maybe it's not on the surface of the cell. Maybe it's just not expressed up there, and maybe it can't really signal until plasticity. But it turns out when she used protocols to just label what was on the surface of the cell, that she sees plenty of MHC on the surface of both dendrites and axons at all ages that we examined. So that suggested to us that MHC is present at the right place at the right time to play a role in synapse formation, as well as later periods of activity-dependent plasticity where it's thought to play a role in elimination. And so we did the typical set of developmental neuroscience experiments. We did loss of function and gain of function to see what the function of MHC might be. And so the experiments are illustrated here. Basically, we plate our neurons and that's the 0 days in vitro. Then we transfect them with siRNAs to decrease surface MHC as synapses are starting to form. We then wait only 3 days, so this is a pretty short period of expression, and then we fix an immunostain those cells for pre- and postsynaptic protein, and I'll show you some images of that in a minute. So there are many different forms of MHC class 1, and we wanted initially to take a sledgehammer approach, and so instead of targeting these, we targeted beta-2M with our siRNAs. And so what that does is that actually decreases the ability of any newly synthesized MHC to be exported onto the surface of the cell. And we did a lot of controls showing that we get about it's not a knockout, but we get about a between 40 and 60% decrease in surface MHC on these siRNA transfected cells. And so when we assess synapse density, what we typically do is in our control or our manipulated cells, we'll label using immunocytochemistry for a presynaptic protein and a postsynaptic protein, and then where they co-localize, we count those as synapses. And so in these experiments, we were labeling for a vesicular glutamate transporter that's at glutamatergic synapses, and this is a cocktail of NR2A and B subunits, antibodies to both, because the antibodies to NR1 are really not very good. And so when we less MHC, and it was higher to the 5% higher. So this was really interesting to us because we were getting these results before Mike Greenberg published his paper on MEF2, and we thought, this is the first demonstration of a molecule that negatively regulates synapse density, and we were presenting it at Neuroscience, and we hadn't figured out mechanism yet, but we were really excited, and then I went to a poster. Many of you probably have this experience, and Mike had his full story and his paper submitted to Science, right? So it took us a while to dot the I's and cross the T's on this, and then we published this in 2011 in Nature Neuroscience. But that, I think, is one of the most interesting aspects of this, just from a synapse formation point of view, is what that tells us is that neurons are actually capable of forming many more synapses than they do. So what you read in textbooks is that neurons, axons get into their target, they form many, many exuberant connections, and only later, during activity-dependent periods, are they pruned back. In fact, there are quite a lot of regulatory mechanisms that regulate that initial formation of connections. It's not just unregulated exuberancy. And so we think that MHC is playing a really important role in that, and so we're investigating that now. So this is a function not just of siRNA transfection in cultured neurons. We also see that in beta-2M knockout animals, and when we quantify synapse density from electron micrographs taken from layer 5 of visual cortex at multiple ages, that synapse density is higher at every age examined. And the developmental profile of this is also interesting because the greatest increase is during this period when synapses are just starting to form. And in these animals, there's this really interesting phenotype of brain overgrowth that would be predicted from these changes in synapses, which is interesting from the standpoint of a subset of autistic individuals having brain overgrowth. And so we're really interested in looking at these animals much more intensively using behavioral assays and in vivo electrophysiology now. Yeah.
In the layer 5 EM synapses, are those specific synapses on the specific dendrite of a certain cell, or just any excitatory synapses?
They're any excitatory synapses. So our first pass was just we took about 100 sections through, so they're not serial EM either. But we wanted to get as much of a survey as possible to see whether synapse density was changing in vivo.
Any sense that there would be differential response if you were looking at, apical dendrites of layer 6 cells in layer 5 versus, any sense of?
It'd be great to know, right? And so right now, you could conceivably do that with siRNA in slice cultures, right? So some of the stuff that I did for graduate school. But the problem is that you don't get such a great knockdown, right? So we're actually generating conditional knockouts of specific forms of MHC, and we'll be using some probably biolistic transfection it'll be a floxed allele, and so we'll put the drivers in through transfection into sparse neurons and then be able to knock it out in those neurons. And so I think that that's probably a better experiment. We don't have the tools yet, but we're going to go into that and start looking at cell type specificity as soon as we have those tools. Mm-hmm.
Is there any other phenotype in knockout myocyte besides brain overgrowth, like changing activity?
Well, let me be clear. the prediction is, and the exciting possibility is that there's brain overgrowth. The only thing that we've looked at is synapse 5. Okay, so there's a lot that you would have to do to actually show that there's brain overgrowth. So no, nobody. In these animals, there are defects in activity-dependent plasticity that the Schatz group has shown. There's a deficit in LTD, which makes sense if you're not getting synapse elimination. So they think that that's what's happening. As far as behavioral assays, they haven't been done, and so we're starting to do that in collaboration with a wonderful behavioral neuroscientist, Jackie Crawley, who recently moved to, Alexei knows her, yeah, she recently moved to UC Davis.
And is there any change in the lifespan?
Not that's been reported. Not that's been reported. So there have been some reports that were really anecdotal in their initial paper, and I think they need to be quantified, so we'll be doing this. Increased ventricular size, which is also interesting, related to some of the things that I'm going to tell you about in a few minutes. Okay, so yes, it's unsatisfying because we need to know a lot more about these animals. Okay, so I'm going to fly through this. So function, we wanted to do exactly the opposite, but look to see whether overexpression of MHC might decrease synapse density. We used the same method, and we found that actually, in fact, it does. And the synapses that we have looked at aren't just changing, the cells aren't just changing synapse density, but they're also changing their function. So Paula Garay, who was a graduate student in the lab, used whole cell patch clamp recording to record miniature excitatory postsynaptic currents on these cells in cells that had decreased levels or increased levels of MHC. And what she found was exactly what was predicted by the changes in synapse density, that if you decrease MHC, mini frequency goes up. If you decrease MHC, mini frequency goes down. But she also found something pretty interesting, which is that the amplitude of these minis was changing, as well as the frequency. And that suggests that synaptic strength is being regulated by MHC levels, and that perhaps MHC might be regulating AMPA receptor trafficking. And so we're starting to investigate that distinct possibility now. So hopefully I've convinced you that studying MHC in the early developing cortex is important, that it negatively regulates the initial establishment of connections. And so I hope that a number of you are thinking, well, how could it be doing that, right? So you know that it goes up onto the surface of the cell. How is it working? And we're starting to get a little bit of insight into this, but not full answers yet.
So is the effect only on excitatory synapse, and what about the inhibitory tone in this? Maybe what you are doing is decreasing and increasing inhibitory tone, and that could explain both amplitude and frequency.
Right. So what we're doing is the effects. So we looked at synapse density for inhibitory synapses, and we also measured miniature inhibitory postsynaptic currents on these cells, and basically see effects both in the same direction. So it limits. So it decreases when you overexpress it, it decreases inhibitory synapse density, and it decreases the frequency, but not their amplitude, okay? And so if you make an estimate of excitation inhibition balance onto these cells by looking at the conduction, basically by estimating that from the minis, you get a really dramatic change in the balance of excitation and inhibition, even though MHC is affecting them both in the same direction. So whether MHC is having a more direct effect on one or the other, we don't know. We do know that MHC is found at these excitatory synapses. We have other evidence that it's found in inhibitory synapses, but we don't know what proportion. Okay. So basically, there are a number of different mechanisms that you might propose for explaining how MHC might be having this role. The first is that it might be binding to an immune receptor, and a number of immune receptors have actually been found in the central nervous system. The T cell receptor that you might typically think of has not been found to be expressed in the CNS by the Schatz group, but there are natural killer cell receptors that are MHC binding partners that have been found in the nervous system. The PRB knockouts, the Schatz group has shown, do have phenotypes and activity-dependent plasticity, and some of those phenocopy the knockdown of MHC, but not all of those phenotypes do, suggesting that there are other receptors. PRB does not seem to be mediating the effects that I described today on synapse density. We've been looking into the Li49 receptors. We have some data suggesting that these might play a role, but there are 13 different members in C57 mice. It's really complicated, and we don't have specific enough reagents to tell yet. We hope to get those. We also have been entertaining the possibility that maybe MHC molecules bind to other synaptic proteins at the synapse that they wouldn't typically be thought of binding to. And so a grad student in the lab has been using mass spec to pull out some of these proteins, and we're just starting to identify what those are and starting to test those functional interactions in the process of looking at synapse formation. What we do know, though, is what effectors after MHC gets up onto the surface might be. And so we published a paper in Journal of Neuroscience a year ago, or a year and a half ago now, showing that once MHC comes up onto the surface of the cell, it works in an activity-dependent manner to activate calcineurin, which then activates MEF2 transcription factors to negatively regulate. And so if you remember back to the list, to certainly interact. And so we're starting to plug in some of the other synapse-limiting molecules to see whether all of them go through a similar pathway or whether there are multiple pathways for limiting synapses. So rather than tell you about that paper, I want to spend the last few minutes that I have, the last little section of this talk, and tell you a little bit about why we think this is so important and especially relevant for disease. And so I think there's a lot of basic science looking at synapse elimination and synapse limiting the formation of connections that MHC. But I think one of the real excitements in the field when Carla's group found MHC in the brain is that led to the possibility that maybe a peripheral immune response could alter brain development or function by altering the expression of MHC molecules in the brain. And so, as being mediators of the immune response, it may be that they're mediating an immune response during typical brain development. So if that was going to be true, you might imagine that there would be a lot of evidence suggesting that there might be a link between strong or abnormal immune responses and neurodevelopmental disorders like schizophrenia and autism. And so I want to summarize a little bit of that because this is an increasingly compelling list of associations, I think. So one of the first things that came up about 4 or 5 years into us studying MHC, just because it was interesting, was an association, a genome-wide association study that showed that one of the few sets of genes that are associated across the population with schizophrenia are found within the MHC complex. Okay, there are a lot of genes in there, but they certainly are immune genes. And it's turned out that that's been replicable and that at least part of that large signal is MHC class 1. And that data should be coming out soon. There are also some recent genome-wide association studies that also haven't been published that are finding MHC at a genome-wide association level to be associated with autism as well. So epidemiologically, there's been evidence for many years it's been accumulating, suggesting that an immune response, at least ongoing immune dysregulation, is associated with these disorders. And so patients and relatives have an increased incidence of autoimmune disorders for both individuals with schizophrenia and autism. There's evidence of neural inflammation in postmortem tissue from both diseases. There are reports of increased plasma cytokines that are ongoing and that may correlate with different states of disease in schizophrenia and autism. And there's reports of altered brain cytokine levels in postmortem tissue from both. And then finally, there's this really interesting association of maternal viral infection at the end of the first trimester that lands the mom in the hospital with an increased incidence of offspring getting autism and schizophrenia. So this is probably the most compelling environmental risk factor for both of these disorders. And so we started to wonder whether maybe MHC might be downstream of these immune responses in mediating this kind of association and this increased incidence for schizophrenia and autism. And so the first thing that we did was to start to look to see whether the cytokines that are altered in the brains of individuals with autism might regulate synapse density and MHC in our neurons. And so I'll go through this relatively quickly. Basically, we used our same culture system and we applied 6 different cytokines that have been found to be altered in the brains of individuals with autism. We applied those to our cultures, waited just 2 days, and then measured synapse density. And we found that every cytokine altered synapse density, which is sort of a problem, right? Because as a scientist, you want to find one that does something really well, other things that don't. And so we found that all of them do. Some of them increase synapse density relative to controls, and others decrease it. And interestingly, they also alter MHC, and they do so in the direction that's predicted by their change in synapse density. And so for those cytokines that increase synapse density, they decrease MHC, and the cytokines that decrease synapse density increase MHC. So my graduate student, when we first got this data, we were really excited. We thought, okay, now we're onto something. Now we just need to look to see whether these changes in synapse density require the changes in MHC. But then we thought about the experimental design, and we got a little too thorough. And what we did by getting too thorough was to look at the dose response curves. And we were talking about this a little bit earlier. So there are some cytokines like TNF-alpha and MCP1 that have typical dose response curves where they plateau at a greater concentration. They can either increase or decrease synapse density. There are others, though, like IL-6, that have a bell-shaped dose response curve, and others like IL-1 beta that have a sigmoidal dose response curve. So at that point, we thought, all right, how do we know what density is relevant, what concentration is relevant, right, when we're doing our studies? And could it possibly tell us anything about what's happening in the brain if we alter one cytokine at one concentration? What's happening in these is that if you have a combination of changes in these cytokines, it's going to be a combination of ones that increase and decrease synapse density and a combination of what's happening in doses, right? So at that point, we had to step back and say, well, we need to go and measure this in vivo, and we wanted to do that not just in typically developing mice, but we wanted to do it in mouse models that were relevant to these disorders. And so we turned to this risk factor, this maternal viral infection, and we thought, let's go and look at the literature and see if there are mouse models of maternal viral infection, and then see if we can't collaborate on those. And it turns out that this was a huge field that had been established for many years. There is a number of mouse models of maternal immune activation, some where they expose the moms to flu, others where they mimic that by injecting mice at mid-gestation with poly-IC, and there are several others as well. These were originally developed by Joran Felden's group. They've gone on to be used extensively by Urs Meyer and Hossein Fatemi, and of course also by Paul Patterson, who was at Caltech and unfortunately passed away about a year ago, really. So what all of these labs have found is that when you inject the moms with poly-IC mimics a viral response. They get sick, they lose a little bit of weight, and then they go on to have litters that really look pretty typical. They're of normal size, they're not sex skewed. So everything looks fine until you measure the behaviors in the adults. And so in these behavioral assays, what people have found is that there are a whole series of altered behaviors, what you might find in schizophrenia and autism. There's decreased PPI, decreased social behavior, decreased working memory, and impairments in all three core symptoms of autism. So we thought, well, maybe these are mouse models of disease, maybe they're mouse models of peripheral immune regulation. We really wanted just to look at these to ask the question of whether a peripheral immune response might alter the expression of immune molecules in the brain in offspring. So the first thing that you would think peripheral immune response are immune signaling molecules called cytokines and chemokines. And these are molecules that are thought, at least early in development, to cross the blood-brain barrier potentially, and they could potentially lead to long-lasting effects in the brain. And so in collaboration with Paul's group and a wonderful student in Paul's lab, Elaine Shao, they sent us brain tissue from offspring at a series of different ages. And so we looked over time at the levels of 23 different cytokines at the protein levels using a Luminex 23-plex chip. And so I'm just going to summarize the data here. This is a heat map showing those results. They're results of levels of 23 different cytokines from the frontal cortex of the control compared to the maternal immune activation offspring at a number of different ages of those offspring. So at birth, during the period of synapse formation, during plasticity, and in adulthood. And at this point, this is in frontal cortex now. So white boxes mean that the cytokine levels didn't change between groups. Red boxes mean that they were statistically significantly increased, and blue means that they were statistically significantly decreased. And the depth of the color indicates the fold change. So there are two things to take away from this. The first is that this maternal immune activation absolutely does lead to long-lasting changes in the expression of immune molecules in the brain of offspring throughout development and into adulthood. The second thing to take away is that the changes are not always in the same direction. They're not always the same cytokines, and they're not always in the same direction. And this is something that's really important if you're going to study these kinds of processes, because cytokines are a set of signaling molecules that maintain homeostasis of the immune response. They are a network that are going to affect each other in positive and negative feedback loops, and that are going to undergo dynamic changes. And so you can't just look at one cytokine at one time point and have that tell you what's happening in the process. So the pattern that we saw here was really interesting. Cytokines are generally up, and a number of pro-inflammatory cytokines and chemokines are up at P0. But then what happens during brain development is that they fall through the bottom. They really decrease in their levels. And then they come back up at P60, which is consistent with the neural inflammation phenotype that people have reported in these mice and also in schizophrenia. And so certainly maternal immune activation is altering cytokines. And so we wanted to know whether they alter MHC and connectivity in the brain. And so this was work that was published, again, about a year and a half ago. And there were a couple of different ways that we looked at MHC on these neurons. The first was to take neurons from the frontal cortex of newborn pups from either moms that were injected with saline or moms that were injected with poly-IC. And then Micah Estes, who's a student in the lab, stained those for MHC. And so the white dots are clusters of MHC on these neurons. And you can tell immediately that there's a big difference. These are representative dendrites. And so there's lots more white dots here on the MIA neurons. And when you quantify that, you can see that the density of surface MHC on the surface of these cells has doubled, which is a pretty big effect. We verified that with flow cytometry. And so we then wanted to see whether this increase in MHC would lead to the predicted decrease in synapse density. Nobody knows, nobody's reported whether there are synaptic changes in the brains of these offspring.
So what was the control on your genetic experiment?
Those are offspring from saline-injected mothers. Okay? So what we found was that there is a dramatic decrease in synapse density in neurons that are cultured from offspring from saline-injected moms versus offspring from poly-IC injected moms.
How about using your knockout mice, the MHC class 1, to see if these interleukins don't change?
So we can't do that because we can't mount an immune response in the moms. We tried that. Yep. So this is why you need the conditional animal. And in fact, what we want to do is we want to overexpress it in an inducible way. So let me just finish, because I know that there's some people who have to leave, and then we can do questions if you don't mind, because I want to just get to one other little point and summarize. So basically there's a pretty dramatic decrease in synapse density. This is the largest effect that we've ever seen in synapse density. And through a series of rescue experiments, what we found that the change in MHC is necessary for this MIA-induced decrease in synapse density. And so basically, if you just look at this graph, if you quantify synapse density in control neurons, it's about here. If you if you culture the neurons from the MIA model, synapse density is decreased. But if you normalize MHC levels by transfecting some of those cells with the beta-2M siRNA, you basically rescue your synapse density. Okay, so what that means is without affecting anything else, when you bring the MHC levels back down to normal in the MIA neurons, they don't have a change in synapse density, which starts to give us a pathway for what we think is happening. So maternal immune activation is altering brain cytokines. It's then altering MHC on neurons. In data I'm not going to show you, we know that it also taps into that MHC-MEF2 pathway that I told you about, and that then leads to changes in synapse density. And what we still need to know is a lot, which is true for newborn pups from this model, but we don't know whether this MHC pathway mediates any changes, if there are any, in synapse density or synaptic function in these brains over time. So we need to look at much later ages. And we don't know whether this pathway, even if it does mediate synaptic changes, whether it has anything to do with the autism or schizophrenia-related behaviors in these offspring. And so there's still quite a bit to figure out here. And so our working model is really that maternal immune activation leads to changes in cytokines that are really dynamic and change over time. Many of these cytokines work through individual receptor families, but they converge on a common set of intracellular signaling molecules called the JAK-STAT signaling pathway. And so we're looking into this to see if these might be a central hub where there's a switch that then regulates MHC expression. This is known to be a pathway that regulates MHC expression in immune cells, and that then is the readout for these diverse changes in regulating synapse density and synapse development. So we think that immune molecules on neurons and possibly glia may mediate both genetic and environmental contributions to psychiatric disorders like schizophrenia and autism. And one of the things that we're starting to think about now, though, is whether this actually is a good model for disease. And when you think about a mouse model of psychiatric disorders, that becomes kind of nebulous, right? I mean, how does a mouse have schizophrenia? How does a mouse have autism? And if we're going to really go down the rabbit hole of figuring out exactly what the cytokine receptors are that are altered in the brains of these offspring, I want to know whether they're going to be relevant for us to use as targets for developing new diagnostic tools or drug discovery. But the only way that I can know that is to know that those receptors that I find in the mouse are going to be relevant to primates. And so we've started and I've run an interdisciplinary collaboration between 8 groups actually at UC Davis over the last 3 years. And this is a really innovative program that was started by the to promote interdisciplinary research. And I should tell you about it because it led to just some incredible sets of projects across the university. But basically, we're using our mouse model of maternal immune activation. Melissa Bauman and David Amaral have established a macaque model of maternal immune activation. And we're collaborating with the Cam Carter, who's a cognitive neuroscientist who studies at-risk kids for psychosis. And he watches a subset of these kids as they go through their first break and end up with a schizophrenia diagnosis. And we're collaborating with immunologists to look to see whether there are peripheral biomarkers that are consistent across models. And we're developing some, tapping into some of the in vivo imaging that was initially started in humans is now being used for small animals. And we're trying to adapt this to young mice as they're developing to see if we can watch what's happening to the brain over time. And so I'll just show you some interesting results with the rhesus, and this is my last slide. And that's basically that in this model, this maternal immune activation model in the monkeys, it's basically established in a similar way, but there are all sorts of autism and schizophrenia-like behaviors that have been identified in these animals and are just being published now by Melissa Bauman, whose group has done this work. And these include atypical social interactions, decreased vocalizations, and also stereotypies, repetitive behaviors. One of the 2 things that we've done here is that we've validated the model for its relevance to schizophrenia by using PET imaging, where we've seen increased striatal dopamine and cortical inflammation. This increased striatal dopamine is the hallmark of psychosis in humans. And so what this is telling us is I think that these animals, this is a great model, a really valid model for schizophrenia. And so now in my lab, we've gone on to start to look to see whether the changes in immune molecules that we see in the mouse brain might overlap with what we see in the non-human primates. And the results have been incredibly surprising. I thought maybe we'd find one out of a bunch that didn't overlap. We might find one that did. What we find is that out of 23 cytokines receptors assayed so far, only 5 receptors decrease, and 5 receptors are altered, and they all decrease in the brains of the non-human primate model. And those 5 receptors almost completely overlap. If you look at their type and their function, they absolutely completely overlap with the 6 that are altered in the mouse brain. And so we think that across species comparison is going to give us some confidence as to which ones to go for to start to develop some new treatments we hope that will target immune dysregulation and new diagnostic tools that we might be able to use to pull out a subset of humans that may have a neural immune-based basis for their psychiatric disorder. So I told you about a lot of work. It's been done over a large number of years in the lab with some amazing people. These are some of them. This is Brad Elmer, who did the MEF2 work, Lee Needleman, who did the localization of MHC, Paula Garay did almost all of our cytokine work. Marianne Glenn started us off on this whole adventure into immune molecules in the brain. Micah Estes is a current graduate student who's working on the MIA model and looking at the role of IL-1 beta. Xiaobo Liu was our EM collaborator. We had a number of really awesome collaborators on this, Paul Patterson, of course, and Ted Jones. And then a lot of this project was actually funded by private foundations. And so those of you that are junior faculty, you really can run research programs and go off into a completely new area by getting these private grants. And they're small, but they're worth it because you can piece them together and start a whole new area of research. And that's exactly how I was able to do this. So I'm really grateful to these foundations. And we've actually, we're continuing our MHC work funded by NINDS, but we've actually just turned that collaboration into a successful NIMH Conti Center at UC Davis to study the cross-species MIA model. So thank you. I'm a little bit over, and I appreciate your attendance.